Okay, so today we're going to be covering what most people consider to be the bane of immunology, is B-cell receptor recombination, which it's actually not that bad. It's just one of those things where it's a lot of little things to remember. And I did want to also have, I will put up a study guide for this, but also I'll put up, some people want to see kind of more movies or animations that would give you kind of a better feel for what's going on with Vdj recombination. And I was hoping to be able to show like one or two of those, but the internet's down. So I'll just put up a page that has the links to the animations, because those kind of help. And next week I might bring a slight activity to do that'll kind of give you a little more hands-on way to remember this. And I may need more than just today to cover this, but or we'll try and get through it, but then kind of review it next time. So basically we've talked about kind of the role of B cells and T cells and adaptive immunity. So how did these are all within at least my lifetime. We've discovered how this works, and how do we have such diversity in our receptor repertoire, our repertoire of what we can see. And even early on, people were doing experiments with just antigens, so they were just putting foreign substances in and looking to see if they could raise an antibody response against it. And just by doing that, they were kind of throwing out numbers, and initially they were thinking, based on what they were seeing, that we were probably producing at least like 10 to the 7th different possible combinations of receptors, B cell receptors and T cell receptors. And that number was actually even off from what it actually is. But still, even based on that, people were trying to figure out, well, how do we have such diversity? Because at this point in time, what we knew was, genetically speaking, one gene produced one protein. And so how could we have all of these different T cell receptors? So early on, obviously, one of the first theories was wrong, was that one gene produced one T cell receptor, and we just had massive numbers of genes. And they argued that was probably evolutionary. There was an evolutionary advantage to having that, and therefore we would dedicate all this genetic material to giving us this great diversity. Turns out that that's not true. And when you really came down to it, there was more diversity than we had genetic material to produce. And so, so how do we get this diversity? But with a limited amount, I mean, we have a lot of DNA, but still, when you look at the numbers, it's still a limited amount of DNA and genetic information to produce this really extensive library or of T cell receptors and B cell receptors. So the mechanism used by B cells to produce this repertoire were figured out in the 70s. And they say with elegant experiments, they were pretty cool experiments, because this was before we had sequenced the genome. This is before we could sequence, and this is before. So this is back when we were just really, you really had to work to figure something out. And so it was kind of cool how they figured it out. And if we have time, I'll go over it. I'm not going to be on the exam, but basically how they figured it out was they took a B cell, they took a plasma cell that was making antibodies. So they said, okay, well, this cell's making what we want, what we're curious about. And let's get another cell that's very much like this B cell that's not making antibodies. And let's get the DNA from them. So they isolated DNA. Let's chop it up with the restriction enzyme and into pieces equal. So should be cutting them in the same places, because these restriction enzymes are fairly restrictive. And then let's let's run them on a gel to see where the different bands run, and then let's probe them and we're gonna label these probes from our plasma cell and not from the other cell. And so they basically were able to find that there basically was there were differences between the beta cells. So there were differences in the chromosomal DNA. There were differences in the length and in the location of these little chunks that they chopped out of the chromosome. And so that they kind of threw the theory out that we have one gene making one protein and said, well, that something else is going on. We're having some recombination events occurring in the somatic cell. And so when I say somatic, I mean adult cells. So these are cells that are cells in the body. These are not stem cells, and these are not progenitor cells. These are somewhat mature cells, and we're having recombination of DNA happening. And at this time we understood splicing and exons and introns and mRNA somewhat, but we had never seen something like splicing out of pieces of chromosomal DNA to make a unique gene. So how do we do it? So we eventually figured out that there are regions in the chromosome, on different chromosomes, that express these receptors, B-cell receptors, and they have these variable regions. And when we discovered these variable regions, they called them variable because there was a huge variety of what you would see out there in this region. And then we had these joining regions and these constant regions. So remember when we kind of covered a little bit of structure of an antibody. Our antibody is made up of constant regions. Okay, we have heavy chains. So here's and then we have light chains. So our antibody is basically made up of identical heavy chains. So these two guys are the same. We have disulfide bonds, and then we have basically our variable regions, and then we have our constant regions. So everything here is constant. And so basically it's that this is where we're getting the variations, and these are going to be what recognize the different antigens. And so we have variability, we have these junctions, and then we have our constant regions. And these splice in a way to where one variable region will be joined to a joining region, and then that will be situated closer to a constant region, and you'll get expression of this gene. And it also, without this recombination event, the promoters for these receptors are too far off. So they're too far from the gene to initiate transcription, and therefore cells that don't have this recombination event occur aren't going to make receptors. So you need this recombination event to occur. And mice, organisms that have mutations in the proteins that help facilitate this recombination, they don't make B cells and T cells because they never start making these receptors. And so that's an immune disorder where basically it gets rid of your adaptive immune response. And anybody who's done research might have heard of RAG mice. These are mice that one of the proteins that helps to facilitate this recombination event are called RAG1 and RAG2. RAG, R-A-G, and we'll have that on the slide coming up. So basically, you get these recombinations within the genome, within the chromosomes, and they will facilitate now the expression of this unique receptor for that cell. So B cells use a group of parts of genes to create different possible antibodies using a recombination. It's kind of like shuffling a deck of cards, giving out different hands. And the regulation of this machinery is tightly controlled so that you only get one receptor. And you only get a receptor if it's, and we try and select receptors that aren't specific for autoantigens for cells. So B cells, again, use these parts to create different possible antibodies using recombination. So not only are there variable regions and joining regions, but there's also, in the heavy chains, we have these diversity regions. And so this is where the term, and you're going to hear it quite often, Vdj. So Vdj recombination is then going to be the mixing and matching. That's like the analogy of the cards. We're going to mix a V with one variant of a V with a different variant of a V with a different variant of a J, and we're going to throw in a couple random nucleotides in between those. And so we're really going to switch things up, and that's where your diversity is going to come from. So let's see. Let's remember that Ig, or antibody proteins, consist of two identical heavy chains and two identical light chains. Light chains for humans can be either kappa or lambda. So not only do you have this recombination happening, but we have genes for lambda, and these are on even different chromosomes. So you could have recombination of the gamma light chain, and that's going to contribute to your So you're going to have, not only do you have this mixing and matching of within the gene, but then you can choose between either the gamma or the kappa light chains. So you can have, but you can't have both. So they're also tightly regulated. So you only either get expression of kappa, so you would have the same kappa, or you would have the light chains from the lambda. So, yeah.
Do you mean on each antibody, or do you mean each person?
Each antibody. So, or each antibody is going to and the percentages are different between these. So some of your B cells are going to have a lambda, and some are going to have a kappa, and it's just when they are developed. It's like 40, 60 or something like that, and I think there's.
So each antibody will only have one light chain, one kind of light chain.
Yes, only one light chain. Yes, and so that's important to know is that each B cell, it's going to develop, and it's going to have this recombination event happen when it's developing in the bone marrow. So in the bone marrow, you have an immature B cell that's not expressing any receptor. It's going to go through this recombination event where it's going to mix and match its variable regions, its diversity regions, its joining regions. And once those get spliced, they're going to bring into close proximity that promoter, and they'll start expressing that. And as long as it doesn't recognize self-antigens, then you're pretty much fixed with just that receptor. If it recognizes self-antigen, actually B cells will get thrown back into a second recombination loop where they can actually recombine again, and if they still recognize self, they'll be killed. If they don't recognize self after that second recombination event, then they'll go on.
Why are they doing second recombination? Why are they doing second?
If they recognize the self-antigen. So if they recognize, similar to what happened with the T cell, how we have positive selection and negative selection in the T cell, you have a similar selection occurring with B cells.
So that's positive or negative? this B cell?
So that's going to be a negative selection. So we're
So
selecting.
you select one for self-antigen gene.
Yeah,
You don't have a self-antigen, they survive. Then go another.
they'll go another round of recombination if they recognize self-antigen. And
Another.
yeah, so well, we'll look at the arrangement of the genes to see that you can actually you can do that. So
they give them a second chance.
So you have two identical heavy chains, two identical light chains, in any given mature B cell. Light chains can be either kappa or lambda, and each set of gene families are encoded on separate chromosomes. Kappa light chain genes include B, J, and C. They don't have that D. Okay, this is where they vary. Mice have 18 of these variable gene families. Humans are similar, have a similar arrangement, but a differing number of B's and J's. So we have lots of different B's, a few different J's, and constant regions. And we'll talk about the constant regions, how the constant region is going to be your isotype, your antibody, what type of antibody. Is it IgM, is it IgG, IgD? So that's going to depend on which constant region you are going to have in your mRNA. But so that's at a different level. So let's continue to focus on Vdj recombination within the B cell receptor. We've got our lambda light chain. So now we're just focusing on this lambda light chain. It pairs. So the first thing that happens, okay, and so this is, so in humans, 40% of the light chains are going to be this lambda type. So that goes back to your question of what percentage. So 40% roughly of our B cells are going to have this lambda type of light chain. And we have about 30 variable genes, so humans, so that's an important one. I'm not going to have you guys memorizing the mouse, but for humans, you have about 30 variable genes are used, and there's 7 basically J constant pairs, 4 functional. So what happens is first you get recombination between the J and C segments. So you first are going to stick together. You're going to choose a C. You're going to basically choose which joining segment you're going to have, and you cut it out between that. And I'll just go over that real quick. We'll go over that in a second. So you're going to have recombination of the light chain, but you're also going to have recombination of the heavy chain. So same thing, except we throw in a D. So we have our, we say B heavy, D heavy, J heavy, and then the C heavy segment. So for the heavy chain, we have 38 functional B segments. Mice have even more. Downstream of the B is the D, and that's kind of the order they are in the genome. And 30, we have more, so we have more diversity in that D. So we have about 30 plus in humans. And then J, we have 4 in mice and 6 in humans. So it's probably important to, kind of remember like 38, over 30 B's, and then 6 J's. And so it's the combination of these different numbers, as you can see, we're gonna have, that's gonna give us a lot of diversity. It doesn't quite get us to 10 to the 11th, but it gets us pretty high just having just that variety where you can mix and match between 38 some odd B's, 30 D's, and then 6 J's. So just that alone gives us a lot of diversity, but it doesn't get us to the numbers that we're seeing. And so there's also some diversity thrown in. When we join these regions, the enzymes that are the repair, basically these are DNA repair enzymes that are going to also throw in a couple extra nucleotides in between, and you're going to get variety in that way. So that really, once you do that, then you really can make a huge array of different antibodies.
I have a question.
Yeah.
So, what's like the functional difference between like variety genes or the variety genes and the diversity genes? Like, wouldn't they kind of be like the same thing? It's just trying to create like.
So how we also differentiate, and we're going to see is you can't mix two Ds together. You can only get a B on a D, and so regardless of what they look like, it's this kind of the sticky end that is associated with it. So the V's are going to have one type of sticky end, and the D's are going to have a different type of sticky end that can stick together.
Each other to create the diversity.
Yeah.
Okay.
Yeah.
Would you say that mice would have more variety gene segments because evolutionarily they come in contact with more stuff than we do?
I don't think I'm an expert enough to speak on that point. I mean, it's an interesting hypothesis. You could look it up.
Yeah.
We're pretty dirty as well, and we live very closely with mice. So if they have,
When you say this mice, is it laboratory mice or just?
Oh, in general, I mean, the laboratory mice are going to be pretty representative. It is as far as genetics of natural or wild type mice, because these are fairly conserved between all of the strains. And so, I don't know, I couldn't speak on as to that because they actually have less variety in the D segment. So you could, in general, humans and mice have pretty similar diversity in their repertoire. It's just mice have more B regions, and it could be maybe they come into contact with more, evolve more closely with things. I don't know.
Would you say that like you said that they kind of complement each other with like how they create diversity? Do they create diversity within like the genes in different ways? Like if mice have more of these, but we have more of like the D genes, then would you say that I don't know. I guess I don't really know how to word my question.
No, I hear you. Is it a different type of diversity, or
Yeah.
do they play a different role? I mean, they are going to be in different locations.
Right.
Within that physical protein, they're coding for, something more middle D-wise and J-wise, but a lot of the structure in these genes, even though I know those end ones, and actually I think the diversity ones that end up coming into more contact usually with antigens than the variable regions, even just based on where they are. So I think that's where they actually started. May have named them the diversity genes because they actually that even though it's not on the tip. So I know when we draw antibodies, we kind of are saying that, well, this is what comes into contact with your antigen, right? But because of folding and whatnot, so if we were to really focus in on these genes, you've got your V. Your V is going to code for this portion of the protein. Your D is going to code for that portion of the protein, and your J is going to code for that portion of the protein, and then all the rest is going to be constant. Region.
So Vdj is the variable, recombination is the variable, right?
Yeah, but usually I think the majority of, because of folding, the majority of antigens actually physically come into contact with most of them with that D region, and I think that's why they call them the diversity region. Don't quote me on that one actually. I'll have to look that one up again. But so the fact that mice might have more diversity here as opposed to here, I don't know if that. And in general, yeah, I don't know. I mean, I'd have to have a statistician really look at it, but there are a lot of mouse antibodies. I mean, just from a practical point of view, you can generate a mouse antibody to almost anything in single human. There's just enough enough diversity to where you can get antibodies are pretty diverse, and you can get them targeted to almost anything. It seems like. So, but I don't know. It's a good question. It's definitely something I know that mouse immunologists look at and think about. But why do we have different? Why do they have so many more than us? It could have been a viral event that basically produced that at some point evolutionarily. So I don't know. So we have these variable region genes number, and so this is the numbers in humans and mice. Don't worry about memorizing mice again. We tend to, I think most of you are going to be more worried about humans in the future, and you're much more clinically oriented, and so really that's what's important. So so the kappa, so our B kappas of our light chain are going to have. So here we say B kappa, so we're assuming this is light chain, 34 to 48 functional. And that's really the only number that matters. We have a couple pseudogenes, open reading frames. So really it's that 34 to 48. And if you just said over 30, you'd probably be right. J's, we have 5 in the kappa, whereas we also have 5 in the lambda. So they probably evolved and then split off, maybe onto different chromosomes. Who knows how that happened? But then you have the lambda B's, we have 33. And then in the heavy chain, we have 38 to 44 B's, we have 23 D's, and we have 6 J's. So again, these are going to be your heavy chain, and they don't write, for D, they don't write heavy because there's only, D is only found in heavy. So the little nomenclature. J, you have to differentiate. Are you talking about J on the light chain? Which light chain are you talking about? D's are only found in the heavy chain, so we don't have any H on it.
So in the bottom number, what do you say? Do we need to remember any of that, or just remember human, how many we be?
It's good. These are good numbers to know for a human. I wouldn't memorize anything else necessarily. Okay, so what are the genes involved, and what are the proteins and enzymes involved in recombination? So RAG1 and RAG2 are really important. And again, anybody doing immunological research, RAG mice were developed, and they were incompetent, and they had an innate immune system, but they didn't have an adaptive immune system. That's because these two proteins are very important for facilitating this recombination. And if that doesn't happen, then you don't get mature B cells and T cells. So these are expressed specifically in our lymphoid cells, and they catalyze the DNA strand breakage. So they kind of hold the homologous regions together, and then we have our TDTs. TDT is going to facilitate the joining, but also they are going to be involved in adding more information. So they'll add a couple nucleotides in there, and that's where you get some more random insertions and variety. We have our HMG1, 2 proteins, they stabilize. So a lot of these are not unique to the lymphatics, or to your B and T cells. RAG1 and 2 and TDT are going to be specific for your B and T cells. Everything else is part of your normal DNA repair mechanism. Okay. So how do we get the joining? How does this physically happen? And so you have these recombination sequences, RSSs, or recombination signal sequences, so this is important to know. So you have certain recombination signal sequences associated with your Bs, and a separate one associated with your J's, and this just depends on whether you're talking about the light chain, kappa, or lambda light chain, or the heavy chain, which ones are on which. But basically, what you have is you have these, and if you look, we have an AC, a bunch of A's, and if you look here, you have a homologous pairing with potentially here on this other guy. So basically what happens is these can anneal together when you separate the strands and anneal them together. They'll meet up with each other. But these have a different, so they found these common sequences that helped them to identify this, but they found that there was always this random non-conserved 23 base pairs in between some of them, and then there were 12 random base pairs inserted in between other ones. And when you look at the DNA, the winding of DNA, they kind of, that clued them into that specific 23 versus 12 was allowing for basically a second spin in the DNA to happen. So these, when DNA folds back on each other, this was making sure that these were matching up to the other because of how many base pairs you had. So that allowed, so if you look at the turning of the DNA, so you get this structure of DNA. What was happening was, and when DNA folds back on itself, that the 23 versus 12 was allowing them to have homologous regions that would meet up. And this is where your RAG1 and 2s come in and basically facilitate homologous pairing of one of these with one of these, and then you're going to get, so here we have our 23 base pair spacer is in red or orange or brown or whatever that is. And so you would have, that's how we make sure that you only get a B matching up with a J and not another B. So you have these RSSs that flank each antibody gene segment. And so each has a conserved, again, monomer and heptamer sequence. And in between the monomer and heptamer lies these spacer
What's
sequences.
a nomer and a heptamer?
So 9, you have 9, so, and then heptamer would be 6. 1, 2, 3, 4, 5, actually 7, sorry. I got one back.
Heptamer is 5, what's it? 7.
7. Hex would be 6, heptamer 7, monomer is 9, that would be 0. So these are conserved. Now, these spacers aren't conserved, but and that's what also clued them in, because they weren't conserved, but how long they was conserved, basically said it doesn't matter what's in there, it's just the spacing, so that you space them out, so you get equal, so you get the right number of turns of that DNA helix, so that they'll match up with these guys.
Okay.
So you have, like, for example, I have, let's say V1, and then you have this RSS with a, let's say, 23 spacer. So we have this 23 on this V, and then we have this J, let's say J, and J1, and you have this J1, and then on this J1 we're going to have the 12 spacer, and when you get a loop where you have green matches up with this orange, and you get recombination that occurs there, and then you splice out anything that's in between it. So if I had a
Do you figure unless you have a 14?
So if I had a V2, I could have a V1 attached to a J1, and then I would splice out. So the V2 would be on here, the V1 would be here, and the J1 would be here. And so you would basically splice out this V2, which would go off and get basically metabolized, and you would what you would end up with is this V1, J1. So basically that allows for basically they will cause these loops or hairpins, and then join the rag1 and rag2 will come in. It will cleave them out. Then other repair genes will come in, add some nucleotides in between. So that space in between them may have a couple random G's and C's thrown in there, a couple G's, a couple C's thrown in there. And so that's going to vary, and then that's going to vary what amino acids are coded. And so you're going to get this huge diversity of potential amino acids that are thrown into your receptors. So you can only have the recombination between a 12 and a 23, so you're going to only be able to recombine like a V with a J as opposed to V with a V. And so you can end up with, here's the example of V3 and J3 are being combined. And then anything in between is going to be like this J1 and J2 are going to get tossed out. And this is where I said that if for example, if this were to recognize self-antigen, then this can go through a second round of recombination where you could have, like, for example, V2 could combine now with J3. Oops, go back. So V2 could recombine with J4, and we could just get rid of those threes, the V3 and the J3. So if these have to go through a second recombination event, you can do that, because you still have information that can get recombined and make a different receptor that maybe now won't recognize self. So your germline DNA will now be, so basically you're now changing your germline. And so what you'll end up with is you're going to end up with, and here's a light chain example, and here would be a heavy chain example, where you would get V's that would then bind to a D and then to a J. And the first recombination that happens is this D and J. So think this whole dance starts with the DJ before you get recombination of B and D. So first you have DJ or rearrangement, and then you get VD rearrangement. And then for the light chains, you're only going to have Vj rearrangement. So you've got this 12-23 rule, and you also have the order of that rearrangement occurs. Okay, so what the players involved are RAG1 and RAG2. RAG stands for recombination activating gene. Okay, you need both RAG1 and RAG2. RAG1 forms the complex with the RSSs, stabilizing it and binding to RAG2. And then numerous other proteins are required, several that are not unique to lymphocytes. They're unique to all cells because we all need to repair our DNA now and then. So you get this Vdj recombination. And how does that happen? You get this basically looping back, and then your rags come in, bind that, leave out this hairpin, which basically forms this hairpin and goes off. And then what you have is you have to get rid of, basically you have these loops that are formed between the 5' and 3' ends. Those get cleaved, and then you add some information and then anneal them together and end up with this variable and joining our variable in J. So the RAG proteins bind to your RSSs, cleave the DNA. Other proteins come into the process, process the hairpin loop that forms, and then your product includes this joint and the leftover is the groove. Okay, so step 1, rag 1 and rag 2 come in. So rag 1 will bind the RSS and catalyze the synapse formation between the B and the J. So then it forms a single-stranded nick at the exact 5' border of the heptameric RSS bordering both the B and the J segments. And then hydroxyl groups attack the phosphate group on the non-coding strand of the B segment. So basically, you get this loop that forms in between the two strands themselves, holding back on them in between the two, and then ligation of the signal end occurs, and you basically get, you end up with your sequence of the signal junction results from the joining of the two.
Do we need to know all this? Yeah.
Do you need to know all of this? You need to know the gist of it, not necessarily the chemistry of it per se. And then what happens is now you end up with, you have to open the hairpin, you cleave off a portion of the hairpin and generate sites for P nucleotide addition, and then you get ligation of the light chain B and J regions. So you have so definitely I would go over this. You're also going to add non-template nucleotides, and red are added. So here's where you're adding some random nucleotides in when you're fixing these guys so that they will match up with each other. And then you basically will repair basically that junction, and now you will have a V next to a J. So you would have like V1 next to J2. And this always goes that the genes are always going to go in the same direction before recombination. Let's see what we've got. And so you have two different, okay, so that's what this is trying to tell us. So we have two different ways that these genes can so now you have even more diversity that come in, because you can take your V, and if you create a hairpin, your V and your J are going to be going the same direction they were initially. But you can also form these genes in a way, you can form them in a way where when you recombine them, you're not getting a hairpin, but you're actually going to have them going now opposite directions. And so you can actually recombine these guys facing the same direction or opposite directions based on how you join them up. And one of the videos that I have on YouTube kind of demonstrates that a little bit better, so that you can see that either you get this hairpin where they're going the same direction, or you get this other conformation where they will actually link going in opposite directions. So that you can get more diversity in there. So the 5 mechanisms that generate antibody diversity are, first, you have multiple genes. So you have lots of Ds, or a couple Ds and a couple Js, which gene segments are put together. So first you have different cards in the deck that you're going to shuffle. Different cards that you can shuffle together to make your deck. And then you have P nucleotide addition. So you're actually templated nucleotide addition between joints, results from asymmetrical cleaving of the hairpin structure. So you're cleaving this hairpin structure, and then when you repair it, you're putting in more information, you're putting in a couple more nucleotides, and so now that's going to be randomly generated, and so now you're getting diversity that way.
What determines what kind of nucleotide we're adding?
It's random. It tends to be more G's and C's, I believe, but there's no mechanism to edit that process. Exonuclease trimming sometimes occurs at junctions, so you lose some nucleotides. So you're adding nucleotides, but you're also sometimes losing nucleotides, so you're not always getting the same length, and so your sequence is changing, your frame, so you know how transcription occurs depending on your codons. So if I throw 3 codons, I'm going 3 by 3 by 3. If I throw that off by 1, now I've shifted frames of reading frames, and so I'm making a totally different sets of amino acids and so basically that random chewing back, sometimes exonuclease activity, random additions will now create basically this huge array of potential amino acids that you end up with. Non-templated end nucleotide addition mediated by this TDT activity. So
What's the thing?
so P and N are the different strands, the different sides. So that's what is sticking off when you cut. So when I cut my DNA, I'm going to have a 5' end and a 3' end, and it's what's sticking off of those ends.
So which one?
I think P is on the 5' cut and N is on the 3, but I'm just going by memory from that. So, by mechanisms generate antibody diversity in IV cells, combination of this diversity, which heavy chains pair with. So now you have the other combination of, yes, we've recombined, we make these different heavy chains, but then what heavy chain goes with what light chain is going to change the shape of that, depending on, so let's say we have our heavy chain as B1, D1, and J1, and he can team up with a light chain that has, let's say, a light kappa that has a V1 and a D1. No, J. Sorry, no B's. J1. But this would be different than one that matched up with the kappa V2 J1. So basically, any combination is now going to change how those shapes, and the shape is what's going to dictate what it binds to. And so now you can have just this huge array of different shapes that you can generate just because you're just changing all these up. So first of all, just looking at combination of diversity, just looking at gene diversity, you're already getting number of heavy chain segments when we mix these different number of kappas that are potentially generated, different number of lambdas that are generated, and then just the combination now of this potential 165 different potential lambdas with 5,658 potential, and then this is not even accounting for exonuclease activity, addition of nucleotides, orientation of the genes. So this is just pure numbers. If I mix these versus these with these guys, then I can get 2.909 times 10 to the 6th potential different combinations. So we're already at 10 to the 6th, and we haven't even added these random cutting back and random addition. So I mean, really, that's where we start to get into the 10 to the 11th realm, and that's not, and we're not even talking about yet about something called somatic mutations that are potentially another round So once, these are all just getting us to our B cell that's expressing a receptor. But when B cells encounter antigen, they go through another process where they go through somatic hypermutation and will actually mutate. They have a mechanism that will induce mutation of this gene further and see if they can get better binding to antigen. So, and that just is even more. So, really, there's really no limit to what combinations we can produce. Allelic exclusion. Okay, so the other thing is so at the beginning I said you can only have, we only have one gene for heavy chain, but we have two different genes for, or two different, even on two different chromosomes. We have light chains, we have the kappa and the lambda, and you're only going to get either a kappa or a lambda. You're not going to get both. And so the process of making sure you only get expression of one is called allelic exclusion. Okay, because you're going to exclude one. You want to express one, you want to exclude the other. And so, because recombination is a very ordered process, so heavy chains are recombining and expressed first. So before even light chains are recombining, we're first recombining our heavy chain. And we make we make a heavy chain, and it's going to be made. We're going to choose a couple B's and D's and J's. We're going to recombine them. That's going to happen. That's going to get expressed and actually presented on the cell surface. And then there's, so you get this functional heavy chain, and it shuts down. So expression of that will shut down temporarily all the recombination machinery. The heavy chain is paired with a surrogate light chain to form a pre-B cell receptor. So you get this surrogate it doesn't have any variety. It's just a surrogate.
What's the word surrogate?
It's a placeholder. It's something that will give, because these heavy chains, they need something to give them some shape, to say, hey, to give them structure. So they'll have this. It's going to look like a light chain. It's just not. It's going to bind there, and then the surrogate light chain will pair with the heavy chain, and the machinery is started up again. The light chain recombination then takes place. So first we're making that heavy chain. We're even throwing in a surrogate maybe to help it keep its structure so it doesn't fold back on itself. And then allelic exclusion ensures that each B cell synthesizes only one heavy and one light chain. So here's the whole process. So first you get your pre-B cell receptor made up of your heavy chain. In this case, it's the mu or your MIGM. So again, remember we have different. Let's see if I got it. I'm just skipping. I don't want that figure. So let me get us back to where we were at. Okay. So we have different like IgM. So we have lots of these B's, and then we have lots of these D's and J's, but then we have C's, and we have Ig, so we have mu, so that goes for Ig and M, and then we have Ig, I think the next one is D, and then we have different all the different constants. And so when you combine your B's, so you end up with your B, let's say your D that you choose, and your J that you choose here, and then your constant region, and then you get all of these constants. So your mRNA will actually initially, and here we have a promoter that's brought in closer, so your message will initially include all of these constant regions, and then depending on which one is spliced out is what your Ig is. So it could be IgG, IgM, IgB. So that depends on which one is spliced out as RNA. So all of what we've been talking about is actually DNA splicing, and then you're going to choose what isotype of IG you have later on once that messenger RNA is made and whether it's spliced out. And that's probably why the majority of our IgM is the most common, because that's that first C fragment. Okay, so just as a side.
Now the first, most common, right? IgG is most common, right?
Yeah, it's most, because it's soluble, it's not in the cell membrane, but like, for example, infants, when infants are born, IgM is very prevalent. So I'd have to look up the specific numbers for you. But that's at first, initially, you're going to have IgM and then IG. I think.
But it's when you first encounter antigen, yeah, you know, so the first response is IgM.
Yes.
But in blood circulation,
Yeah.
they are not prevalent.
Yeah, because eventually you do accumulate more. But yeah, of more soluble proteins, IgGs, yes. But for when you first start making an antibody, your IgM is going to be that first one that you get. And we'll talk about that more later. It's not important now because we're going to talk about class switching in a later chapter. So what is important is just that you know that you're only going to get one light chain. And how does that happen? You have this pre-B cell receptor, and so again, you have a certain heavy chain plus the surrogate light chain inhibits rearrangement first, and then your first is going to induce is this kappa rearrangement. So then we're still selecting, and then you're going to have mu and kappa chains inhibit rearrangement of kappa little 2 and lambda rearrangement. So, and then the heavy chain and light chain, kappa light chains, inhibit rearrangement of this lambda here. And then, so each time, and then what you're having is basically you're forming IGs with the kappa, and then if you have what's called a productive allele, then that will go on, and that will basically be your allele. If it's a non-productive allele, so if it binds to self-antigen or if it doesn't bind to anything, so we still have this positive and negative selection going on, then it'll go on and it'll allow. So it's basically you're first going to get your heavy chain is made. It's going to make sure you're not making anything else. And then you're going to make the kappa, you're going to allow your kappa to rearrange. And it's going to group with your IG, your heavy chain, and if it recognizes self, it's going to say, hey, throw you back into the mix. Then it'll let a second round of kappas. So it'll try again. So again, if you look at, like for example, which picture? So if we have, for example, let's say this first 3-3, so this guy produces a kappa that is going to recognize self-antigen. We say, no, that's not going to work. So we throw it back in, we allow him to recombine again. So we allow him to recombine with 1 and 4 or 2 and 4, and then you end up with a 2-4, and then that will combine with the heavy chain. And if that doesn't recognize self, then and it's a valid allele. If it goes through positive and negative selection, then it'll go off, and that'll be what you have. Yeah.
Do the genes between variable regions, the random assortment between the 12 and the 23 pairs, determine whether the immunoglobulin ends up being the IgM, IgG, IgD?
No, that's a totally separate process. Yeah.
But does it discern what's specified out of the genes, or is it the C clusters that determine?
So it's just those which C you end up expressing is going to be whether it's IgM, IgG, IgD.
Okay.
And that choice is made later on, and that's not made at your DNA level. That's an mRNA splicing event.
Okay.
And so right now, it's all you're getting. And so whether your variable region, it's going to be the same shape on all of those IGs. So it's going to be, let's just say you recognize a ball. And you don't recognize squares or triangles, but you recognize a ball. That same ball is going to be recognized in IgG, IgM, IgG, you know, all those are going to, it's
Right.
that constant region is just going to decide avidity, because based on how many they're going to form, whether it's in the cell membrane, whether it's secreted, those kind of things.
Okay.
And that choice is made later on, and it's not made based on the type of variable region we have, or the type of binding that you have.
Okay.
At least not at this stage, and not at that level. Later on we'll talk about how type of binding may induce different things, or the cytokine milieu will maybe dictate certain things. So you're going to go through a second round, and if that second round works, then that's what you get. If it doesn't work, then it's going to go, and it's going to say, well, kappa failed me, let's go on to lambda. And so now it's going to let the brakes off of lambda and allow lambda now to get tried. And so now you can get the formation of a lambda, and then a lambda is allowed to go through, and if it's productive, then it'll be what's expressed, and if it's not productive, then you go through a second lambda allele, just like you did with your kappa allele. And then if you still get a non-productive allele, then you've gone through the whole process, and that happens, and that happens with T cells.
When you write the mu, mu,
That's going to be the heavy chain.
a mu is that?
Yeah.
I thought a mu is, IgM.
Yeah.
Oh, you just use the IgM like that.
Yeah.
Okay.
That's just that I guess we're saying it's a mu, which is why I kind of was trying to explain that. That's just which one is expressed at this stage. So that's which constant region is expressed. So they're going to call it a.
So it can be anything.
Yeah.
Can be 5 class.
Well, for this process, it's just going to be mu for the selection process. And then once
With
it's
selection process.
so through the whole positive and negative selection process, just like with T cells and B cells, until you get that T cell receptor, until it becomes a mature cell, or an adult cell, let's call it an adult cell. Until it becomes adult, it's only going to be that.
Out of mu,
Yeah.
out of the IgM.
Yeah. Until it becomes mature, and then, until it becomes an adult, and then it can switch.
So that's called a class switch?
Yep. So, so that's how you regulate and make sure that you're only making one or the other, because you're basically allowing them to go in order. First you're making the heavy chain, then you're making the kappa, then you're letting kappa go again, and if that doesn't work, you're tossing out kappa, and then you're going to start with lambda, allow lambda to go twice again, and then if you're not recognizing self, if you have binding, low-level binding, then it's gonna be a productive allele.
So I have another question.
Yeah.
If, everybody got a chance, 2 chances for a light chain.
Yeah.
Right? If recombination, they express, one of the light chain, they just go through, that's finish.
Yeah.
If they don't finish, that means they have 4 chances to
Yeah.
choose the light.
They have up to 4 chances, yes. Or if it randomly is the first V recombines with the last J or whatever, then that's 1 chance. But that doesn't always happen.
Oh,
So no, remember we have like 30. So in the rare event that you get V1, the first one, recombined with Vdj that's not gonna be a
J
question.
is 30.
J, whatever the last J is. How many J's do we have? 6? Is that right? Anybody
5.
remember? 5? Okay, 5, that's right. So we have 5 J's. So if J5 were to combine with V1, then that's it.
Oh, so you have one chance.
Then you would only have 1 chance. But that's only a 1 in,
1 in 5.
possibility.
So 20% chance you only get 1 chance.
So, but for the most part, you're gonna get 2 chances.